Okay, jangan lari Kak. Kalau kau lagi dengan aku seorang ni. Okay, senyap terus. Okay, tak apa Kak. So, for topic 4, we are going to discuss a few of the more famous questions. Okay, so apa yang nak discuss mostly ialah pasal normal dihybrid and linked genes. Okay, sebab dua topik tu memang selalu keluar. Topik yang paling famous. Dan juga the two topics are the ones yang memang students usually have difficulties lah. Okay, figuring out whether the question is about normal dihybrid ataupun they are asking about linked genes. Okay, boleh nampak ni Nuraini ni?
Boleh, sir.
Okay, so let's do one question. Okay, so in one experiment, a purple flower and long grain plant was crossed with a homozygous red flower and round pollen grain plant. The number of progeny of F1 generation is shown in table 1. Purple flower, long pollen, 46. Red flower, long pollen, 39. Purple flower, round pollen, 38. Red flower, round pollen, 43. Okay, so first kali untuk soalan macam ni, you need to figure out whether the question is asking about, aku nak admit sekejap ya, I'm going to admit 1% dulu, alright. Okay, for this type of question, dia kata using P to represent allele for purple flower and L to represent allele for long pollen grain, determine the genotype of the following individual. So first, jom discuss how do we make genotypes. Okay, because kita tengok sifat dia kat sini, they have two phenotype. Okay, genotype ialah dia punya GGLL, ni ialah genotype. While phenotype is the characteristic. Okay, so G besar, L besar, L besar is green and long, for example. So ni genotype, ini phenotype. Okay, dihybrid pula maksudnya ada dua sifat. Okay, so this question is a dihybrid. Dia cakap ada dua, purple flower and long pollen grain. So ini ialah dihybrid. Okay, purple flower and long pollen ialah dua sifat, dua phenotype. So dia ialah dihybrid. Okay, dia cakap pula is crossed with the homozygous red flower and round pollen grain plant. Okay, red flower and round pollen grain plant maksudnya ialah dia cross dengan homozygous recessive. Okay, so kita tengok konsep dia macam mana. Okay, dia ada dua jenis cross in dihybrid. You have either a test cross or a self cross. Okay, a self cross is bila awak ada heterozygous cross dengan heterozygous, and this will give you 9:3:1 ratio. Manakala test cross ialah bila awak cross dengan homozygous recessive. So T besar, T kecil, S besar, S kecil, T kecil, T kecil, S kecil, S kecil. Dua-duanya homozygous recessive. The cross is with homozygous recessive. So this is a test cross which will give you a 1:1:1:1 ratio. Okay, so bandingan ratio untuk self cross dihybrid ialah 9:3:1. Untuk test cross dihybrid ialah 1:1:1:1. So first kali soalan ni saya soalan yang simple. Okay, you kata kat awak, please write down the genotype for the following individual. Purple flower and long pollen grain. Purple flower and long pollen grain. So the biggest mistake yang pelajar biasa buat kat soalan macam ni ialah dia nampak purple flower, dia buat P. Dia nampak long, dia buat L. Ini genotype dia kata. Okay, dia kata ni lah genotype dia PL. Okay, betul ke salah? Betul ke the genotype for purple flower and long pollen ialah P besar L besar? Ni betul ke salah? Ini ialah salah. Salah. Kenapa je salah? Okay, very simply is because if you have to do a genotype, dengar sini elok everyone. If you have to do a genotype ada dua allele. So you mesti P, P, L, L. Dia tak boleh P dengan L sahaja. Kalau monohybrid dia P besar, P besar, P besar, P kecil, P kecil, P kecil. Kalau dihybrid, that means P besar, P besar, L besar, L besar. Right? P besar, P kecil, L besar, L kecil. Okay, P kecil, P kecil, L kecil, L kecil. So you have to write allele twice in a genotype. Dan sebab kita ada dua sifat, that means dia kena ada 4 allele. P besar, P besar, L besar, L besar. P besar, P kecil, L besar, L kecil. P kecil, P kecil, L kecil, L kecil. Right, so now questionnya ialah purple flower and long pollen. So again, 4 allele. P, P, L, L. Besar ke kecil dia? Right, so we know that purple flower, in the question dia dah cakap, purple flower and long grain was crossed with the homozygous red flower and brown pollen. So that is a test cross. Kita tahu purple flower long pollen ni is the homozygous dominant. So dia ialah P besar, P besar, L besar, L besar. Okay, silap. No, I mean that sebab dia dapat result purple flower and long pollen ni dapat 1, 1, 1, 1. Dia mestilah P besar, P kecil, L besar, L kecil. Okay, because we got that from the ratio here. 4, 6, 3, 9, 3, 8, 4, 3 ialah 1 to 1 to 1 to 1 ratio. 1 to 1 to 1 to 1 ratio means that is a test cross, heterozygous cross dengan homozygous recessive. Mendalah ni. That's when you get 1, 1, 1, 1. Okay, so that means that long purple flower and long pollen grain must be heterozygous. P besar, P kecil, L besar, L kecil. Okay. Manakala homozygous red flower and brown pollen, homozygous red flower and brown pollen ialah homozygous recessive. So dia ialah P kecil, P kecil, L kecil, L kecil. Okay. So how do we figure this out? It's based on here, the data that we have here. The data shows that this is a 1 to 1 to 1 to 1 ratio. So this must be a test cross. Dan kalau dia test cross, dia mesti heterozygous with homozygous recessive. Okay, the homozygous recessive ni ialah the red short flower, the red round pollen. Manakala the heterozygous tu ialah purple and long pollen. Okay. Kalau kita kata purple flower and long pollen ni ialah P besar, P besar, L besar, L besar, you will not get the 1 to 1 to 1 to 1 ratio. Itu sebab dia. Okay. So again, you need to remember, first dihybrid dia melibatkan, ada lagi. Okay. Dihybrid involves dua phenotype. Okay. Brown and short. Okay. Tall and yellow. Okay. Green and wrinkle. Okay. Dua sifat, dua phenotype at once. We're looking at two phenotype at once. So dia mesti ada 4 genotype. So let's look at dihybrid itself, right? Dihybrid and monohybrid. Apa beza dia? For monohybrid, we're only looking at one characteristic, red. For dihybrid, kita tengok red and long. Okay, so let's say the genotype for red ada R besar, R besar, R besar, R kecil, R kecil, R kecil. For dihybrid, akan jadi banyak lah. R besar, R besar, L besar, L besar, R besar, R kecil, L besar, L kecil, R kecil, R kecil, L kecil, L kecil. It can also be R besar, R kecil, L besar, L besar, and so on. Okay, so dihybrid means dua phenotype serentak. Untuk dihybrid dan monohybrid, diorang dua-dua ada test cross and self cross. For the test cross in dihybrid, test cross dia yang kita bincang tadi 1, 1, 1, 1, while self cross dia ialah 9, 3, 3, 1. While in your monohybrid self cross, for test cross dia akan dapat 1 to 1. Untuk self cross dia akan dapat 3 to 1 ratio. So ni ialah basic yang kena hafal. Monohybrid test cross will get you 1 to 1. self-cross 3 to 1. Dihybrid test cross will get you 1 to 1 to 1 to 1. And dihybrid self-cross will get you 9:3:3 1 ratio. Okay. So first, settlekan yang tu dulu, dah faham, tak ada masalah. Now we can go back to this question. Okay. Are the genes for flower color and pollen green shape sex linked? Why? Okay, sex linked tu apa? Sex linked is when the genes are located on the sex chromosome. Okay, sex chromosome ialah kalau perempuan dia ialah XX, kalau lelaki dia ialah XY. Okay, XX, XY. Kita tahu that this cross is not a sex chromosome primarily because they are not affected by whether the progeny ialah male or female. Kalau dia punya ratio tu akan affect sebab dia lelaki, sebab dia perempuan, dan baru kita kata dia ialah test cross or self cross. Kalau tak, dia bukan. Okay, how do we know dia bukan? Sebab again, ratio dia cantik 1 to 1 to 1 to 1. Kalau dia follow Mendelian ratio yang kita bincang, test cross 933, test cross 1111, self cross 9331, that means dia tak ada kacau dengan sex link, dia tak ada kacau dengan multiple alleles, dia tak ada kacau dengan codominance, okay, incomplete dominance, semua tu tak terlibat because 1111 is still a Mendelian ratio. So, are the genes for flower and pollen green sex linked? Jawapannya tidak. Ia tak terlibat dengan sex linked. But don't worry, we'll still discuss sex linked question juga later. Okay. So, first kali untuk soalan ni, okay, what you need is you need to understand. Padam semua lah pula. Patah balik. Okay. What you need to understand is very simple. You need to understand when you look at the question, soalan yang biasa kita akan dapat ialah, is this linked gene ataupun dihybrid biasa? Is it linked gene or normal dihybrid? And jawapannya ialah this is normal and not linked. Mana saya tahu? I know the fact that the fact is this is not a linked gene punya question because we have the data. The data here shows that the ratio for this is still 1, 1, 1, 1. Kalau dia still follow the Mendelian ratio, that means that dia bukan linked. Dia just normal dihybrid. If I have to draw this, the question asks me, please draw the genetic diagram for this question. Dia akan dapat P besar, let's do biru hitam lagi. P besar, P kecil, L besar, L kecil, cross dengan P kecil, P kecil, L kecil, L kecil, awak akan dapat 4 gametes. P besar, L besar, P besar, L kecil, P kecil, L besar, P kecil, L kecil. Remember when you're drawing your gametes, you must circle the gametes around. And jangan lupa juga P dengan G ni kena ada kat tepi. This one, you only have one gamete, P kecil, L kecil, and then you cross all of them. And you'll get your F1 generation. Okay, so awak akan nampak P besar, P kecil, L besar, L kecil. P besar, P kecil, L kecil, L kecil. P kecil, L kecil. P kecil, P kecil, L kecil, L kecil. So you get 1 to 1 to 1 to 1 ratio. Okay. So far so good. Boleh terima lagi cerita ni? Ada soalan before we go further yang lagi susah? Tak ada Okay. So ini ialah again kita tahu, we know for a fact that this is not linked gene simply because they are following the Mendelian ratio of 1 to 1 to 1 to 1. Okay. Which will bring us to the next question, the second question. This one, it looks exactly the same as before. the dihybrid question, but this will give you a totally different set of answers due to just one simple fact that is, alamak lambat dah pula internet dia. Apa ni? Mana gambar? Keluar lah cepat. Right. Okay, so this looks like the same question, right? But this is a whole different set of answers compared to the previous question. Kenapa? Let's keep the question first. In maize tu jagung. The gene for colored seed C besar and smooth seed F besar are dominant over the genes for colorless seed and wrinkled seed. Pure breeding strain of dominant and recessive varieties were crossed, and a test cross of the F1 generation produce the following results. Okay. Apa yang dia merepek kat sini? Dia merepek yang you cross a purebred for dominant and recessive allele. What is a purebred? Purebred means that dia homozygous dominant, dia homozygous recessive. Right? So kalau C besar, F besar, cross dengan C kecil, F kecil, so akan dapat dia heterozygous. C besar, F kecil. Then you cross pula that with a test cross dengan C kecil, F kecil. And this will give you the question whether does this question follow the Mendelian ratio or not. Okay, alamak, aku delete semua ni. Lupa pula. Okay, so you can see from the data that they give you, right? Colored smooth seed 380, colorless wrinkled seed 396, colored wrinkled seed is 14, colorless smooth seed is 10. Okay, ini 1111 ke? Jawapannya ialah tidak. If you remember the, betul Nuraini, tidak. Because if you remember from the previous questions, saya kata Mendelian ratio is 1111. Okay, a test cross for Mendelian ratio is 1111. Manakala soalan tadi yang kita dapat tadi itu is not 1111, it's a different ratio altogether. Okay, that means that for this question, this does not follow the Mendelian ratio. Okay, this does not follow the Mendelian ratio. So soalan dia akan tanya, first kali, this is the number one hint and clue whether the question is asking about linked gene ataupun normal dihybrid. The answer is the ratio. Kalau dia follow Mendelian ratio test cross 1111, that is a normal dihybrid. If they're giving you a ratio yang pelik macam ni, ada dua nombor yang sangat besar, the two big numbers are 1 to 1. Alamak, saya keluar. Okay, the two big numbers are 1 to 1, while the two smaller numbers are also 1 to 1. So dia 1 to 1 and 1 to 1 instead of 1111. That means this is actually does not following the Mendelian ratio, and thus meaning that they are actually linked genes. Apa tu linked genes? Linked genes maksudnya ialah instead of having the two genes on different chromosome, katalah gene A dekat chromosome number 5, the second gene is at chromosome number 8, the two genes are on the same chromosome number 4. A dengan B dua-dua kat sini. Okay, they are on the same chromosome. Itu maksud linked gene. Okay. Okay, so this again, question ni mesti tanya, they always ask if it's a linked gene or not by asking you, does it follow Mendelian ratio? Okay, that's why you need to memorize the Mendelian ratio again for test cross monohybrid 1 to 1, self cross monohybrid 3 to 1, self cross dihybrid 9331, test cross dihybrid 1 to 1 to 1 to 1. And this is a dihybrid test cross. Dihybrid test cross patutnya bagi awak 1 to 1 to 1 to 1 ratio. But this question instead gives you 1 to 1 besar and 1 to 1 smaller numbers. So this is not following the Mendelian ratio. Not following Mendelian ratio. Okay. So maksudnya apa? Maksudnya dia tak ikut Mendel. of 1 to 1 to 1 to 1. Basically, awak tahu jawapannya kenapa? Jawapannya ialah because they are not linked. Because the genes are linked on the same chromosome. Itu dia jawapannya, simple je. Okay, but how do you explain this? How do you explain why is it not following the Mandela ratio? Awak cakap benda yang sama je lah. Basically, ayat dia ialah you just say that the genes for the color of the seed and the genes for the texture of the seed coat, okay, the genes for colored seed and the genes for smooth seed tu, they are actually linked on the same chromosome. Okay, so jawapannya ialah satu markah, awak akan dapat satu markah from just saying that the genes are located on the same chromosome. Ini satu markah. Ini satu markah. What about another mark? Okay, where does the other mark comes from? Jawapannya dua markah. The other mark comes from the fact that you have to add another point that and crossing over occurs. Crossing over occurs. Okay, so draw a genetic diagram for the test cross of the F1 generation. Okay, so let's discuss the genetic diagram. So pisahkan kalau sex link, kalau link gene dengan normal dihybrid apa beza dia. Kalau normal dihybrid, we're just going to do C besar, C kecil, F besar, F kecil, cross dengan C kecil, C kecil, F kecil, F kecil. Okay, but ini dihybrid dan ni link. For link gene, awak dah tahu, you know this again, bukan daripada lukisan, bukan daripada awak punya penghafalan. You know dia punya lukisan ni ialah link gene based on again the ratio. Okay, based on again the ratio yang kita ada kat sini. So from this ratio, awak tahu dia link gene. So bila awak lukis, awak kena lukis format yang berbeza. You can't do C kecil, C besar, C kecil, F besar, F kecil. Instead, you have to draw C besar, F besar, slash C kecil, F kecil. Okay. C besar, F besar, slash C kecil, F kecil, cross dengan C kecil, F kecil, slash C kecil, F kecil. Okay, that's how you show that the genes are linked through your genetic drawing. Okay, so again, something that students suka lupa, again, not because they forget, but because they don't know that the genes are actually linked. So because what they remember, kalau nak lukis heterozygous or C besar, C kecil, F besar, F kecil. So that's a problem because if the genes are linked, you need to draw it a different way. So if it's heterozygous, it should be C besar, F besar, slash C kecil, F kecil. Okay, one common mistake that happens also is students know that the genes are linked tapi dia pergi buat macam ni pula. C besar, C kecil, slash F besar, F kecil. This is also wrong. You can't do this. Tak ada, tak wujud C besar, C kecil, slash F besar, F kecil. Dia mesti C besar, F besar, slash dengan C kecil, F kecil. Manakala for the homozygous recessive, dia C kecil, F kecil, F kecil. Ya, ada orang nak masuk. Hilang dah. Right, tak apalah. Teruk sangat ke internet korang ni? Terkeluar, termasuk ni ha. Ni Fatin Aina sekejap keluar, sekejap masuk. Kesian dia. Okay, terima kasih sebab kembali Fatin Aina. Berusaha juga tadi internet tu. Okay, so now let's draw the gametes, right? Untuk gamete, C besar, C kecil, F besar, F kecil will get you 4 gametes. C besar, F besar, C kecil, F besar, C besar, F kecil, and C kecil, F kecil. While C kecil, F kecil will give you 1 gamete, C kecil, F kecil. Okay. And the ratio yang awak akan dapat bila awak cross semua ni ialah again 1, 1, 1, 1. Ni ialah memang that. Okay. But for link gene, C kecil, slash C kecil F kecil slash C kecil F kecil will get you one gamete C kecil F kecil, while C besar F besar dengan C kecil F kecil sepatutnya bagi awak dua gamete sahaja, C besar F besar, C kecil F kecil. But if you go back to the question, it actually shows you four results, bukannya dua. Why is there four results? The answer is very simple. Sebab even though the genes are linked, even though C besar, F besar ni are linked and C kecil, F kecil ni are linked, you can still manage to produce hybrid gametes which is C kecil, F besar and C besar, F kecil. But not because of Mendel punya ratio, bukan sebab law of independent assortment, but because of the process that happens at every meiosis process. Dekat prophase 1, the chromosome can undergo crossing over. Okay, so kalau CF besar dengan CF kecil crossover, sometimes dia boleh tertukar dia punya allele and dia dapat juga C kecil F besar and C besar F kecil. And when they cross, you still get 4 genotype offspring. Sama juga macam normal dihybrid. But what's different? The difference is that the ratio is not the same as Mendelian ratio. In Mendelian ratio, you will get 1, 1, 1, 1. Okay, tapi dalam ni walaupun again sama juga 4 gamete juga 1, 2, 3, 4 sama je macam normal dihybrid 4 gamete, but the hybrid gamete ni tak banyak because again it's not due to independent assortment, but it's due to crossing over, and crossing over is a rare process that happens during meiosis. Okay, so when they cross, the hybrid phenotype will be smaller in numbers compared to the parental phenotype. What is parental? What is What is parental and what is hybrid? Awak tengok kat sini, parental mak bapak yang kita kahwinkan dia ialah heterozygous kahwin dengan homozygous recessive. Okay, homozygous recessive. Kita tahu dia kahwinkan coloured smooth seed dengan colourless wrinkled seed. Okay, kalau anak ini dapat tu, the offspring that they get is similar to either of the parent. Dia sama dengan mak dia, colourless wrinkled, ataupun dia sama dengan bapak dia, coloured smooth. Kita kata dia parental trait. But if the offspring is a combination of the parent, okay, bapak dia coloured smooth, mak dia colourless wrinkled, anak dia coloured wrinkled, kita kata anak dia tu hybrid. Okay, kalau bapak dia coloured smooth, mak dia colourless wrinkled, dapat anak colourless smooth, kita kata dia hybrid. Okay, contohnya kita ambil awak lah. Kita ambil awak sebagai contoh. If your mum is short and your dad is tall, and awak susah nak kata, mak awak right, if you say that the parent of that person is tall and dark, manakala mak dia ialah short and fair, okay, if the offspring is tall and dark or short and fair, kita kata dia parental. Okay, so tall, dark, short, fair, ini ialah parental genotype. Tapi hybrid ni ialah kalau dia ialah a combination of the phenotype from the dad and the mom. Kalau dia ialah tall and fair ataupun dia ialah dark and short, okay, ini kita panggil dia hybrid genotype. Okay. So this will take us to the next question, which is calculate the distance between the two genes and show your calculation. We can calculate distance of the genes because again, based on the ratio that we get, the ratio that does not follow the Mendelian ratio, kita tahu that the genes for color and texture is linked. It's on the same chromosome. Mungkin kat sini dengan kat sini. So we can calculate the distance between the two genes by looking at the number of hybrid recombinant dan juga parental. Okay, the recombinant hybrid again, recombinant ialah yang gabung mak dengan bapa dia, is the key to getting the distance between the two genes. How do we get the answer? Formula dia simple. Okay, formula dia simple. You just take the number of recombinant, 14 dengan 10 ni, 14 tambah 10 bahagi dengan total of everything. 14 plus 10 bahagi total semua. And that is your distance. Okay, tapi tak cukup. First again, awak ambil 14, the recombinant 14, tambah dengan 10, recombinant lagi 1, bahagi dengan total semua, which is tambah-tambah semua, akan dapat 800. Okay. Lepas tu, you have to times it by 100 pula. Times it by 100. And then the answer that you will get is 24 bagi 8 ialah 3. Okay, 3. 3 salah. 3 map unit is the unit you need to use. Mesti ada unit macam kira matematik, macam kira kimia juga. 3 cm, 3 mm. But untuk ini, 3 map unit, atau we can also use a different unit which is centimorgan. Okay, bukan C kecil M kecil, C kecil M besar. That's how you calculate distance. Okay. You need to recognize recombinant daripada data tu, divide it by the total number of everything, and then times it with 100. So again, how do you recognize the recombinant? Recombinant ialah yang gabungan mak dengan ayah dia. Okay. Atau, if you can't figure it out, jangan risau. Kalau awak tak boleh nak figure out, oh mana satu yang recombinant, mana satu yang parental, saya bagi awak clue yang lagi senang. First, kita tahu this is not linked because dia tak follow Mendelian ratio. Mendelian ratio is always 1, 1, 1, 1. This is not Mendelian ratio, dia ialah 1 to 1 and 1 to 1. The clue yang paling besar kat sini, saya dengar elok. The clue yang paling besar warna satu recombinant ialah recombinant ada 2 lower numbers. 380, 396, 14, 10. Yang kecil ialah 14 dengan 10. Dua ni recombinant. Because recombinant is a result from a very rare crossing over. Nombor tak banyak. Dan dua nombor yang besar kat atas, 380, 396, itu parental. So any question that comes out, awak jangan pening kepala, tak faham saya cakap apa. You just need to remember that the two smaller numbers, itu recombinant. Bahagi dengan total dah 100. Selesai. You will get your answer. Okay. Campur dua yang kecil, bahagi dengan total everything, time 100, you will get your map unit. Okay. If you can't understand the concept, at least understand the formula. Senang je. Terlalu senang untuk digunakan. Two recombinant, the two lower numbers. Tambahkan, divide by everything, time 100, you will get the 3 map unit daripada jawapan awak. Okay. So saya harap from this small demonstrations ini, everyone can understand how to differentiate between normal dihybrid dengan linked gene. The answer is in the ratio. Kalau awak tengok bilangan 4 yang dia bagi tu, 1, 2, 3, 4. They give you 1, 2, 3, 4. Pak. Kalau ratio ialah 1, 1, 1, 1, then this is they follow Mendel, and this is dihybrid. But if they give you 4 phenotype and ratio dia ialah 800, 800, 30, 30, 1 to 1 and 1 to 1, this does not follow Mendel ratio, and this is linked gene plus crossover. That's all we need you to understand when you're looking at the question. Okay, that's how you differentiate whether dia link gene ke tidak. Itu soalan famous topik ni, betul? I'm sure a lot of you have been asking that question when we started discuss topik 4 awal SEM dulu, mesti ramai yang tanya macam mana nak kenal link gene ke tidak. The answer is again in the ratio. Kalau tak 800, 800, 800, 800, 1, 1, 1, 1, dia tak follow Mendel. Dia tak follow Mendel, dia link gene. Boleh everyone? Okay. So let's go to another type of question. Unfortunately, we only have an hour. Nak bincang semua subtopik tak sempat. I'm taking a few questions yang suka keluar peksa. So this link gene favorite. Okay, link gene is a favorite. They always try to go back to link gene. Harap-harap this time pun sama. Because again, link gene soalan dia sama je every time. They will ask you, does it follow Mendelian ratio? Jawapannya tidak. How do you know? Again, you look at the ratio. And then they will ask you, why? Why doesn't it follow Mendelian ratio? Then you can answer, oh, because the two genes are linked and crossing over occur. Dua markah. And then they will ask you to either draw ataupun dia akan suruh awak untuk calculate distance. Both again very easy to do except drawing lah. You have to practice your genetic drawing. Okay, it's not hard, but if you don't practice, you can have room for mistakes. Okay, so let's look at the last question that I have prepared, question 3. This is yang saya cakap tadi, mesti ramai yang akan nak tanya. X-linked disorder. Okay, X-linked disorder, meaning that the genes are located on the sex chromosome. Ayat yang sama, ayat yang lain maksudnya sama. X-linked recessive disorder located on the X chromosome, linked to the sex chromosome. Semuanya maksud yang sama, iaitu dia punya gene tu dekat sex chromosome number 23. Kalau jantan, XY. Kalau kakak, XX. Okay. So what is X-linked recessive inheritance? Maksudnya, again, is something you can just find in your notes, but maksud dia ialah genes of the disorder is located on the X chromosome, okay, which cause the phenotype to be expressed in hemizygous male and homozygous female. Apa maksud dia? Maksudnya macam ni, Abang Kakak. If you look at a normal crossing, let's take a monohybrid yang simple monohybrid, right? Monohybrid yang simple, if it's not on the sex chromosome, any chromosome can do one. We have 23 sex, 23 chromosome, mana-mana pun tak apa except for the number 23, except for chromosome number 23. Apa yang awak lukis, awak just lukis R besar sihat, R besar R kecil sakit, R besar R kecil heterozygous sihat, R kecil sakit. Okay, sihat, sakit. Laki ke perempuan sama. But if the genes are located on the sex chromosome, you can't use this drawing of genetic diagram. Awak kena lukis X. Okay, and this is where the problem comes from. Kalau perempuan, is actually the same concept but just a different drawing. XR besar sihat. XR besar, XR kecil sihat. XR kecil sakit. Boleh lagi setakat ni? Kalau perempuan tak ada beza pun dengan normal kromosom, just kena buat X dia. XR besar sihat, XR besar XR kecil sihat, XR kecil sakit. But the problem is with the male. Kenapa dengan laki? Laki, we don't have two X chromosome, we have X and Y. Besar masalah dia. X and Y is not homologous, maksudnya the genes that you can find in X, you cannot find in Y. So kalau nak buat X, H besar, R besar tak boleh. Sebab the Y chromosome do not have the allele for R. So apa yang awak dapat? Awak akan dapat XR besar Y sakit, sihat. XR kecil Y sakit. R besar R kecil tak wujud because you can't make it. You can't make XR besar R kecil because Y chromosome tak ada R. Okay, so untuk lelaki is 50-50. Ada kau sihat atau kau sakit. XR besar Y sihat, XR kecil Y sakit. But for female, dia still macam biasa lagi. R besar sihat, SR besar R kecil sihat, SR kecil R kecil baru sakit. Okay, so when we say about female carrier, ini maksud dia. SR besar SR kecil, dia sihat but they carry the recessive allele that can make you sick. Okay, so let's do a question. Marry a hemophilia carrier. Baru bincang dia what is a carrier. Marries John, who has normal X chromosome. Okay, using a suitable symbol, draw a Punnett square to show the possible genotype and phenotype of their offspring, including the ratio of hemophilic offspring. Okay, so the symbol kita akan guna ialah H lah, senang. Okay, H senang nak guna. Dia kata Mary is a carrier while John is a normal X chromosome. Kalau Mary tu carrier, dia sihat ke dia sakit? Dia sihat. But she carries the hemophilic gene. Ini sebut X, H besar, X, H kecil. Manakala John is a normal male. Okay, dia kata John tu normal. Carries a normal X chromosome. Maksudnya dia ialah X, H besar, and dia Y. Okay, tak payah pening kepala. Kita tak payah fikir dia carrier ke apa. Dia sebab carrier lelaki tak wujud. Dia mesti sakit atau sihat sahaja. Okay. Kalau perempuan susah. Kalau perempuan kita kata perempuan ni dia sihat. So dia ni SH besar ke SH besar SH kecil. But the question tells you that she is a carrier. Senang lah kerja awak. SH besar, SH kecil. Okay. So you have to do a Punnett square. Punnett square ni maksudnya awak kena buat table. XH besar, XH kecil, XH besar, Y. Okay, so ni XH besar, XH besar, XH besar, XH kecil, XH besar, Y, XH kecil, Y. Okay. So this will be the offspring of Mary dengan John kalau dia kahwin. Okay. Kalau dia kahwin, ini ratio dia. Apa dia ratio dia? XH besar, XH besar, besar. XH besar, XH besar is normal female. XH besar, XH kecil ialah carrier female. XH besar Y, normal male. XH kecil Y, hemophilic male. Okay. So soalan dia ialah dia tanya awak, what is the ratio for hemophilic offspring? Dia tak tanya lelaki ke perempuan, dia tanya anak-anak iaitu apa ratio hemophilic dengan sihat. Okay, so from this ratio kita tahu ada 4 yang boleh dapat. 3 sihat, 1 sakit. So awak punya ratio ialah 1 hemophilic and then kepada 3 normal offspring. Ataupun awak boleh kata 1 hemophilic, 1 carrier, 2 normal offspring. Okay, so ini ialah when you have to answer question that is genes linked to the sex chromosome. Kalau genes tu link to the sex chromosome, you need to draw X and Y. So again, topik 4 ni masalah besar dia is students, they know, they read a lot but they don't know when to use which. Okay, sebab tu kita jumpa in normal dihabit tiba-tiba ada XY. Dalam sex chromosome tiba-tiba dia buat link gene. So you need to know the type of question and the type of answers that you are required to understand. Kalau dia bagi awak data ada 4 yang kita bincang itu, is either dihybrid or link gene. Based on the ratio, awak tahulah dia link gene ataupun dihybrid. Untuk sex chromosome, they will always say link to the sex chromosome, link to the X chromosome. Okay. Barulah awak mula buat XXXY. If they have no mention whatsoever in the question pasal sex chromosome, pasal X chromosome, then you don't do anything about it. Okay, the question usually will tell you whether awak kena guna sex chromosome atau tidak. Dia yang cakap the genes are linked on the sex chromosome. The genes are located on the sex chromosome. The genes are on the X chromosome. So kalau awak tak dapat ayat tu, then jangan guna XY. Jangan guna XY in your answers. Sebab ni awak akan salah lah. Awak akan salah. Yang ramai pelajar yang tak sure bila nak pakai, bila dia tahu. Ada yang buat X, ada yang biasa cakap D besar, ada yang XD. So you need to know when to use which supaya awak tak akan membuat kesilapan biasa. Again, this question, if I'm not mistaken, is 9 marks in your exam. Topik ni banyak markah. So you have to be careful, and the only way that you can be sure and confident on answering the examination on this topic ialah very simply to do your PYQ questions, because we have selected the questions yang suka keluar, the type of questions yang sama almost every time dia keluar. So kita dah bahagi dah. Kita dah bagi awak supaya awak nampak the type of questions yang suka keluar. And based on that exercise on your PYQ, sepatutnya you shouldn't have any problem doing your exam if the same question comes out. InsyaAllah, soalan dia ialah takkan lari daripada yang kita dah belajar. Okay, so before we stop, do you have any questions regarding this topic so we can discuss before we go? Ada request ke apa ke? Nak tanya personal sekarang?
Saya nak tanya,
Ya Kak.
apa tu kalau nak tulis formula distance tadi kan yang
Ya.
tadi tu yang saya dah ajar, itu wajib
Ya.
ke?
Wajib apa Kak? Formula dia?
Wajib tulis formula ke? Boleh je terus kira dia punya recombinant tu.
Bagus soalan awak Kak. Bagus. Okay, kalau tanya saya, always write down the formula. Okay, sebab kadang-kadang ada markah. Dia ialah benda yang simple Kak. Kadang-kadang kita buat ni ada markah, kadang-kadang kita buat tak ada markah. So kita buat je lah, kod ada markah. Okay, it's as simple as that. Okay, it's as simple as that. Baik tulis. So write down, kalau kat sini contohnya kan, so tulis ni. Bagus Kak soalan awak tu, very good question. Alang-alang kita nak tulis jawapan kita tulis dulu formula dia apa. So formula dia ialah number of recombinant bahagi total of offspring times 100. Lepas tu baru masukkan 14 tambah 10 bahagi berapa, 600 tadi tu, darab 100. I think it's 800. Dapat 3 minit ini. So, sama juga dengan topik 5 nanti pun, my advice will always be just write down every formula before awak kira. Just to be safe. Just to be safe, sebab kalau ada markah awak tak buat, rugi. Kalau awak buat tak ada markah, tak apa. Tapi kalau awak tak buat, tiba-tiba ada markah, then you will miss mark. So jangan nak fikir, nak buat ke tak nak buat. Buat je, tak ada masalah. Dia tak akan potong markah sebab formula awak.
Okay, thank you sir.
Sama. Ada lagi soalan nak tanya? Asal ramai. Tadi tak ada orang. Itu ada 14 orang dalam ni ha. Mana semua orang datang? Okay, any question? Kalau tak ada question, we can stop here. And harap-harapnya membantulah kelas ni dengan tujuannya memang saya tahu topik ni besar. Banyak yang saya tak discuss, but I decided to just focus on link gene dengan normal dihybrid sebab ini part yang memang student suka tanya. So, students suka confused, dapat soalan tak tahu ni link gene ke tidak. So, I hope by discussing it a lot dalam session ni, yang masuk dan yang akan dengar session ni in the future from our recordings, boleh nampaklah dengan jelas kalau question tu macam ni dia link gene. Kalau question macam ni dia bukan link gene. Itu yang saya nak fokus more. Also added a bit about sightseeing juga, sebab itu pun sama juga. Selalu tiba-tiba entah kenapa soalan tak minta keluar XY. You only use XY if the question says so. Question cakap is sightseeing, pop, baru kita keluarkan XY. Okay, so kalau tak ada soalan, we can stop here. Thank you everyone for listening. Okay, saya doakan semua mendapat keputusan yang diharapkan, yang cemerlang semasa PSBB yang akan datang nanti, insyaAllah. Okay, so kita tutup dulu sesi kita ni dengan tasbih kafarah dan surah Al-As. Okay, terima kasih semua. Boleh bersurai.
Thank you, sir.
Sama-sama.